In the name of Allah, the most beneficent and the most merciful. Assalamu alaikum wa rahmatullahi wa barakatuhu. Dear students, I welcome you once again in Pakistan International School Types virtual learning system. I hope you all are well and fine. Dear students, this is biology class for second year students and this is our lecture number 55 and chapter number 26 we are studying nowadays. Dear students, in this chapter, we will talk about tissue culture and other very interesting topic regarding biotechnology. This is actually the technique of biotechnology, like fermentation, like GMO, genetically modified organisms formation, and like some other techniques like we have seen like DNA sequencing and DNA, all the stuff which we have studied before that is related to the tissue culture. Tissue culture is actually growth of a single cell or group of cells in a glassware or in vitro condition when we grow and manipulate or increase the number of cells from a single cell, and that single cell can be from any somatic cell or germline cell that can be, but that all the growth and manipulation will be out of the body. Out of the body means that is in vitro conditions in the labs. So that is called a tissue culture that has to be done in artificial medium under aseptic condition. Like all the stuff and material must be aseptic and free from all type of pathogens, bacteria, and other microorganisms. Many somatic plant cells, including some fully differentiated types, like differentiated cells. What type of these? Like mesophyll cells, leaf mesophyll cells, especially. So these mesophyll cells are having complete, like intact nuclear plastids and mitochondrial genomes. Like plastids, like chloroplast, also having their genome. Mitochondria also having their own chromosomes and genes, and nucleus is also having it. So all type of genome must be present over there, and they have the capacity to regenerate into whole plant. So that is used in tissue culture, and that term that is called totipotency. Totipotency is actually the Latin word, and that is totipotentia. Potentia means ability, capacity, or power for all things, like those cells which can have the ability to divide and produce all type of differentiated cells of the organism. That is called totipotent cells. For example, we have discussed about a leaf mesophyll cells, and also spores and zygotes are also having totipotency. So there are plant tissue culture and animal tissue culture methods which are followed in biotechnology. Like in animals, tissue culture is also formed, and plant cells also. So plant tissue cultures includes two major methods. So for example, that is in vitro growth, and that is in the form of callus, and suspension cultures also. Suspension cultures means we do not need any type of substratum, and they can move anywhere in the medium in any direction. So the initial part of that plant which is used for a tissue culture purpose, that is called explant. And that explant is like single cell culture that can taken from the shoot, that can be taken from the root. So we can get somatic and embryo culture, we can get meristem culture, we can get anther culture, haploid production, protoplast culture, somatic hybridization also, embryo culture, as well as ovule and ovary culture can be made. Like any type of part of that plant can act as explant, like the part or the cells or the tissue of that specific area can be taken, and that behaves like explant. So it may be complete organ like seed, leaf, or twig, or a single cell like protoplast cell of that part. So what happened, we take that part and we get that tissue sample and we grow into different nutritious medium. Nutritional medium, when it is given to that, like there are hormones, there are nutritions for that type of cells. The cell started to grow. When the cell started to grow, it gets give a mass of the cells that is called callus. Callus formation is done, that is the next step of that. And after that, that callus is separated into like a single cell culture. Like from this mass of cells, some cells are taken and grown into different test tubes or flask or anywhere in the glass medium with the nutritional medium. And then every cell, every mass of a little mass of cells started to grow. And then they are made as plantlets or the young plants, and then they are going to be implanted or shifted to the separate we can say test tube or to the separate pots for further um, we can say further growth. So from there the plants are just shifted to the fields for growth. So in very less time we can get lots of copies of the same exact same plants which are having the same genome. So this is very very important as well, because nowadays we are having variety of different type of organisms. So in tissue culture we can use genetically modified organisms also, and we take that gene, because that gene or the tissue which are having the modified in DNA in that, and that will grow into the similar plants which the parent plant was. So we can have the variety of different type of fruits, vegetables by tissue culture method. And it means that in very short and very less time we can get a great yield of the required character of phenotype. Then if we talk about animal cell culture, so animal cell culture techniques, um, in that the cells are removed from animal, and we have talked about plants. So from the animal to in the and we shift them to in the favorable environment. So for animal cell culture, the cells are taken from the organs for experimental um, of an experimental animal for further studies also, and for the growth. So the cells can be obtained by previously made cell line or from direct from the organism's body. So in that the animals can grow only to a limited generations. That is unlike plants. Plants can grow into unlimited generations, or from tissue culture the plants can be made into full organisms. But here in animal cells, so we can get just limited generations, even in the best medium, nutritional medium which we are given to the animal cells. So growth of cells depends on the sources. Which type of sources? Like if we take neuron cells, so we know that neuron cells don't divide and they do not grow. So like so if we take examples of fibroblast cells, so that can divide to some generations. So that is these are the best cells which we are going to take for the animal cell cultures. So other thing is that like animal tissue culture is used mostly because that is also the limited generation, grow to up to the limited generation. So we can get for recombinant DNA technology, we can use it for gene manipulation or genetic manipulation. It means that like to grow some the same genes as well, and we have to check the control of the genes which are present in the body. The other one for the cancer research, that is very, very important to get this like animal tissue culture, because we can grow these animal cells into the medium up to certain limits, and then we can use them for the next research and identification. So we can use them for production of vaccines, monoclonal antibodies also derived from animal tissue culture. Like tissue culture is used for making the monoclonal antibodies. Monoclonal antibodies actually, those are the antibodies which are made by same type of immune cells, and they are so much specific for only um, specific, we can say specific or one type of antigens. So these antibodies are like monoclonal. Monal means same, clonal means clone, like one type of immune systems are used to make these monoclonal antibodies. Pharmaceutical drugs are also formed by animal cell culture. Okay, now we'll see some methods of animal cell culture. So there are some techniques which are used for that. Number one is encourage dependent, and number is encourage independent. So some cells which can propagate in monolayer. Monolayers means very thin layer of a single celled layer that is attached to cell culture and to the cell culture vessel or to the substrate in which they are proliferating or they are growing. So they grow for the limited generations. It means when the nutritional medium or that the tissue culture vessel is like its boundary has been finished, so these cells just like they are having the limitation up to the boundary of that vessel or that container, and these cells, they are adherent to the vessels or the container, and they are having their finite growth. It means a finite proliferation, like they cannot move out of that medium. So that is called finite cell line also. And for example, the cells derived from any type of tissues. Next one is encourage independent technique. In encourage independent technique, we use suspension cells. Suspension cells means the cells uh, which are present in our body, and they're suspended in all the body, like in we can say in the blood, in water medium. And so they are not adherent to any type of source or any type of substratum. So they grow for unlimited generations. So they are called continuous cell lines. And in that, we can just take the hematopoietic cells, the cells which are going to use to make immune cells or the blood red blood cells. as well as malignant tumors. Malignant tumors are the type of tumors cell which just move from their place of origin to any part of the body via blood. So by these two type of techniques, we can say that encourage dependent cells are having the limited generations and encourage independent cells are having the unlimited generations in tissue culture, in animal tissue culture techniques. Now we'll see transgenic organisms or the organisms which are transformed or whose genetic material is transformed while adding or deleting some part of the genes or DNA. So these are called GMOs also. So these are the free living organisms in the environment that have a foreign gene inserted into them are called genetically engineered or transgenic organisms. So they were, I think there is no need to speak more about the transgenic organisms, that how they are formed, because that is the basic technique of biotechnology. So the first organism which was transformed or whose genetic material is changed, that is called bacteria. Those are called the transgenic bacteria, and they are produced in 1978. In 1978, it was produced when human insulin is going to be formed by this one. So we'll talk about first of all transgenic bacteria, and they are genetically modified bacteria that carry the gene from other sources and are used for the production of desired gene product at large scale. So this is the basic organism which is used in most of the biotechnological techniques, and there we also use a yeast and bacteria mostly. So these are also called G GMOs, genetically modified organisms. So most transgenic bacteria, they reproduce every 22 minutes. So they will get very less time to make the large copies of proteins which we require as a result. They produce a host important substances such as insulin. So insulin was the first material which was derived or which was taken from the transgenic bacteria. Other growth hormone, blood clotting factors, and all these like such type of hormones and medicines were produced, are going to be produced nowadays by transgenic bacteria. So the first transgenic bacterium was Escherichia coli, which is used to produce human insulin. And in human insulin, human proteins are all human insulin, except that human proteins are also going to be formed by transgenic bacteria. Then plant growth, for plant growth, there are some, like we can say, to improve that, we also use biofertilizers. These bacteria are spread in the plant's roots to just for increase their productivity. For removal of environmental pollutants, that is also very important and like aspect of transgenic bacteria, like they spread in the place where there are pollutants, and they just destroy all those pollutants, and we can say that that is helpful in controlling the pollution. Then other process, we extract metals from low grade ores by transgenic bacteria. Now we'll talk about transgenic plants. So firstly, it is occurred in France and USA in 1986. So what happened at the time? Tobacco plants were engineered to be resistant to herbicides. So very important um, like important aspect of biotechnology for like those plants. If we just transform these plants from their original genome to the transformed genome, which are herbicide resistant, or we can say like sorry, not herbicide resistant, actually, which are resistant to a herbicide, are improving the nutrient profile of the crop. So that food crops include resistance to certain pests, diseases. So like if the pest is attacking, so it increases it decreases the crop yield, and can damage the crops. And there is environmental hazards also if we use pesticides or herbicides in place of this one. So reduction of that is also used for reduction of spoilage and resistance of chemical treatment. That can also be, then it means we use less chemicals in place of this. If we use these transgenic plants and we change their genetic material and we grow those plants, so there is no need to just use herbicides, use to insecticides, to pesticides. So these chemicals are at the time, then we can avoid them. And also, if we just talk about non-food crops, so they include the production of pharmaceutical agents and biofuels also, like for the production of, they increase the degradation and decomposing of that and like that material by which we can use them for biofuels. So they will help the production of biofuels. Other industrial useful goods as well as for bioremediation is also be done through transgenic plants. So you can have like different varieties of corns nowadays which are colorful and healthy also. So now we will talk about transgenic animals. The term transgenic animals refer to an animals in which there has been a deliberate modification of the genome, responsible for inherited characters. So these genomes has to be changed in that, and like in contrast to spontaneous mutation, deliberately the genome is going to be changed. So you can have a very like interesting example on in the form of diagram, like these mice. We do experiment on that, and then we have changed their genetic material. That's why they are having the genes which can make the inflorescent light in the absence of like darkness. So we can just find these mice anywhere. So these GMOs or genetically modified animals, they are having like great importance and purposes, including enhancing yields of milk and meat production, like enhancing yields of these organisms, like cows and sheep, and like that buffaloes, and increasing resistance to diseases. So when we just inject these genetically modified genes in their genes, so what they do, they are disease resistant, so they do not get any type of pathogens and disease. They also, that is these animals are going to be used for producing drugs. So the animals which produce are used for producing the drugs, they are called transformers. And transformer, like from pharmacy, like from the medicines. So for example, very good example of this is the sheep. In sheep, we take the gene alpha 1 antitrypsin that is used to treat emphysema in human beings. Okay, now we will see technology and healthcare. We know a lot about this technology and how this is helpful for the human's health. But here we'll talk about some specific things also, like the role of biotechnology in the treatment and diagnostic of diseases, how they are used. They are used for the development of vaccines. So vaccines can be biotechnology can be used in three different ways in development of vaccines. Number one, that is separation of a pure antigen using a specific monoclonal antibody. Like by biotechnology, we form monoclonal antibody, which is specific to just those immune system or antigens which are present over the specific pathogens. Number two, synthesis of an antigen with the help of a cloned gene. So, um, like, um, we will just prepare that type of antigen uh, in more in number with the help of a cloned gene. We first clone that gene which produce that antigen. Next one is synthesis of peptides to be used as vaccines. So we will we can synthesize or prepare that type of peptides, like some type of proteins which are used as the vaccines in human body or other animals also. Very important technique of biotechnology is gene therapy. So gene therapy, what type of technique is this? Actually, it is the technique that uses genes to treat or prevent diseases. So that allows doctor to treat disorders by inserting a gene into the patient's cells instead of using drugs or surgery. So in the next times, upcoming times, in place of surgery and drugs, we can inject the required gene or healthy gene in place of um, like ill gene or disabled gene to treat any type of disease. So there are certain methods to having the gene therapy. How does it happen? So there are method is one in vivo method or technique, and the other one ex vivo. In vivo means vivo comes for the living organism's body. If the experiment is going to in the organism's body, that is called in vivo. So normal gene is deliberately directed into the gene by certain type of vectors. Okay, so when we inject the genes or healthy gene in the body, we use some sources. These sources are called vectors, and these vectors nowadays we are using a retrovirus, adenovirus, herpes simplex virus, etc. To we take out their like pathogenic DNA, and we inject our healthy part of the DNA or gene into that, and then we inject through injection into the required area of the cells. You can see here in vivo procedure, these genes are injected into the virus, and there is also the lipid nanoparticles are there. So these are called nanoparticles because they are very small, smaller than the bacteria. So they are injected into the required area, for example in the liver, and so there they are injected, as well as they are injected as we will talk about in the next slides about cystic fibrosis by inhalation or by injecting into the body in any specific area or cells. For ex vivo, genes are inserted into the cells outside of the body, then these transgenic cells are again implanted into the body. We take out the required cells, so extract stem cells or progenitor cells. So we take out in the lab and just keep them in the required medium, and then we add we add the required DNA or RNA in that in those cells, and we grow them for for manipulation. We just keep them for some time into the lab, and after that we inject again back into the required area of the body. So that is ex vivo uh, procedure of gene therapy. So gene therapy is very, very important, but not in all the diseases. But inshallah that now we are hopeful just we can use this procedure for lots of other diseases which are inherited. So here we can see the role of gene therapy for cystic fibrosis. Cystic fibrosis is actually the disease in which a defective gene, or the gene which is called cystic fibrosis transmembrane conductance regulator gene, like it regulates the transmembrane conductance. So that is defective here. And if it is not properly functioning or not present, so what happened that the lungs and digestive system can be affected. Actually, what happened, this gene CFTR, that regulator factor that regulates the production of mucus in the respiratory system as well as in the digestive system, wherever the mucus is formed in the body. So that mucus, that it attracts the water and regulates the ions and water content in the mucus. So mucus becomes thin. That is the normal. But if this gene is not working properly, so the mucus becomes sticky and thick due to not regulation of ions and water in the body. So that's why when the mucus gets stick and thicky, so all the organs which produce mucus, their mucus gets thick like this. This is the open respiratory tract, but if you see the lungs, this bronchiole that gets clogged with this thick mucus, and the airways becomes blocked. So by that, lots of diseases are like this is a disorder in which lots of symptoms and the person can face. There can be the air passageway blockage, so there can be difficulty in breathing, as well as liver can be blocked, pancreas, like glands, and they can also be blocked by this type of disease if the gene CFTR is not functioned properly. So what is the role of gene therapy here? Like when we use a gene therapy procedure for this person, so we give some sort of liposomes. We give inhalers, and in that inhalers that consisting of vesicles, those are called liposomes, microscopic vesicles, which spontaneously form when lipoprotein are put into a solution, have been coated with the gene needed to cure cystic fibrosis. So in that, these liposomes, they are having the healthy required gene in that, and that is covered by the lipoprotein. So that is given by inhaler, and the person is injecting that one, so that will function over there. That is in vivo gene therapy. So the lungs cell will just absorb these type of genes, and these genes just when that will make the link with the DNA which is present in the cell. So they start to replicate, and they will do all the mitosis and common process. So that healthy gene will replace the unhealthy gene or the defective gene. So that is given by aerosol spray, by which the person can inhale that liposomes by inhalers into their into his lungs. That can also be done by ex vivo gene therapy. The cells from the lungs or from any place that is removed, and then add transgene and selected modified cell transgenes over there, and select the modified cells which are modified. Now modified means the transgene becomes a part of the old DNA part, and then they are again injected into the required required area or the specific area. The next we will see scope and importance of biotechnology. In that we will talk about technology, biotechnology that is also said to be technology of the future or technology of tomorrow. So in that, now biotechnology is very, very helpful. Now we are going to the new era of biological computers, in which our DNA is used to make such type of computers, and biochips also. So these are the miniatured laboratories, we can say. We can do very less time with the help of these laboratories, which can perform hundreds or thousands of simultaneously biochemical reactions. And these biochips enable researchers to quickly screen large number of biological analyses for a variety of purposes. And that is also very, very helpful in detection of bioterrorism agents also. So this is very helpful technique, which is new nowadays. We can also having biofertilizers like we use microbial inoculations into the roots of the plants, and then these microbial inclusions, they help for the better growth of the plants, and we then there is no need to use chemical fertilizers in that. Very, very important, the next coming era is of nanotechnology. As we have talked about in cystic fibrosis and gene therapy, like liposomes or the vectors which are having the viral vector, which are having the transgenes in them, they are formed as nanoparticles, and that is used in the drug delivery or drug delivery procedures. There are some concerns about genetically modified organisms or that procedure of the biotechnology, because that is always not helpful. There is the problem of disposing of all spent microbial biomass, where we have to dispose of and how it is to be done. That is alarming nowadays, because this industry is growing, and there is lots of wastage of microbial biomass. There can be toxicity of allergies which are associated with microbial productions, like the organisms which use them and which produce this type of products. So they can have the allergies with the toxicity or the chemicals which the microbes produce. Then increasing the number of antibiotic resistant pathogens, that is also going to be increased, like antibiotic resistance in the organisms that is going to be developed. So that is also the problem for the scientist. Pathogenicity of GMOs to infect humans and others also, like pathogens, like if it is used by certain organisms, humans and other plants and other organisms, so that can be the pathogens for them, and that can cause the different diseases. And the next one is prevention of contamination or mutation of processed strains. So if we use these type of products, definitely that can be mutate some other like when these strains or these microbes are getting mutant and contaminated. So how can we prevent them from these all contaminations? This is also the problem. So these are some concerns and some problems which the scientists are facing now. They are going to find out their solutions, and they think about them deeply. But overall, if we talk about biotechnology, that is very, very helpful in upcoming centuries. Dear students, by this we have completed our chapter number 26, that is about biotechnology. And by this topic, we have finished our biology syllabus for grade second year. So because up to the reduced syllabus, our last chapter is not included in that. So last, yes, last chapter is not included. So we are just up to here, up to the gene therapy and all these solutions and concerns. So I hope you understood it and found informative and interesting also. Just keep revising, and now because you are having not so much time for like delaying your studies. So uh, wish you all the best. Thank you. Allah Hafiz.